too for people to join?
Let's go ahead and get started just so we can keep on time.
Okay, good afternoon. Welcome to the ARO seminar series. Our first session is titled Hereditary Deafness Conundrum. Is an noncoding variant naughty or nicely in disequilibrium? My name is Inna Belyansova, and I will be a moderator for today's webinar. I'm a staff scientist in Dr. Friedman Laboratory of Molecular Genetics at the NIDCD NIH. I work with Tom for more than 20 years already, and I'm MD PhD with cell biology background, and my interest is in functional characterizations of the deafness genes discovered by our laboratory and also gene therapy. I'm very excited to introduce our speaker for you today, Dr. Thomas Friedman. Since 1996, Dr. Thomas Friedman have been the chief of the Laboratory of Molecular Genetics in the intramural program of the National Institute on Deafness and Other Communication Disorders at NIH in Bethesda. For the past 26 years, the focus of his research lab at the NIDCD has been the identification of variants of genes associated with human deafness and syndromes that include deafness, as well as mouse models to study inner ear pathophysiology. For the previous 18 years, beginning in 1978, Tom was on the faculties of the Zoology Department, College of Natural Sciences, and also the Department of Pediatrics and Human Development in the Medical School of Michigan State University. There, he served as a director of the graduate program in genetics, and this program was interdepartmental PhD granting program. And following completion of his PhD as a graduate student at the University of Michigan, Tom was postdoctoral fellow in the biochemistry laboratory at the National Institute of Mental Health at the NIH, where he studied galactose metabolism in cells from galactosemic patients. His dream after completing his postdoctoral experience at the NIH was to one day return to the NIH at Bethesda campus and focus like 100% of his time on human and mouse genetic research and the training of the highly diverse group of upcoming scientists and future ARO members. So I'm happy to tell you that Dr. Friedman Laboratory contributed in collaboration with other labs and by itself to the discovery of about 25% of all known non-syndromic deafness genes. And 18 of total of 76 recessive deafness genes identified to date were discovered by Dr. Friedman's lab. So before getting started today, I would like to review a few useful tools that you can use throughout the presentation. The tools are located at the bottom of your screen. And to keep questions from distracting our presenters, let's use the chat function directed at myself as a moderator. So you can either send your questions to everyone in the chat, or you can select my name and direct the questions to me as a moderator. So any questions that are, all questions will be addressed by me at the end of the presentation to the speaker, and any questions that are not addressed during the discussion will be answered by the presenter in the follow-up email. So there is a closed captioning for this meeting, and it is available for you. It's also at the bottom of the screen. You can see CC there. This is closed captioning, and you can enable it. And this session is being recorded and will be made available for additional viewing. And we hope you will also participate in our post-webinar evaluation so we can gather the feedback. Okay, lastly, if you having any technical difficulty in the chat, there is a link for assistance, like the email headquarters@aero.org, and you can see it already. So, and now, without further delay, I'd like to introduce to you Dr. Thomas Friedman. The floor is yours, Tom.
Thank you very much for that lovely introduction, Nina. Two disclosure statements. We have a created with Ionis, and Ina has a collaboration with Akuis. So the outline for today includes 3 parts of a talk and what might be a megalomaniacal challenge to ARO investigators at the very end. So I want to start off with some definitions, some background information, and the goals of the Laboratory of Molecular Genetics beyond what Ina just spoke about. And then there are 2 stories that I want to tell you. One is I've labeled DFNB39, it's recessive deafness segregating in a lot of families, and DFNA27, which is a dominantly inherited progressive deafness that was segregating in just one large family, and then this challenge. So in the title of the talk, I use the word non-coding, and I want to try to clarify what I mean by a non-coding variant. So the human genome has 26,564 genes as of last week, but there are still some genes that are yet to be identified, that haven't been annotated and haven't been noticed. We're confident of that because we keep uncovering some novel genes. So most encoded proteins and a variety of RNAs are encoded by these genes, which are composed of, in total, of about 233,000 exons, and additional exons are still being discovered in the genes that have been studied for a decade or 2. But taken together, all these exons comprise only 2% of the human genomic DNA. The other 98% is sequence between exons and sequence upstream and downstream of a gene. So a non-coding variant can be located almost anywhere in that other 98% of the human genome that's not exons, or in the 5' or 3' UTR of exons, and I'll explain what that means now. So I've drawn a schematic of a hypothetical gene that has 3 exons, excuse me. So there's a transcription start site for a gene, and then there's non-coding sequence called 5' UTR, untranslated region. Then translation of the message begins, and I've drawn 3 coding exons, and they're separated by 2 introns. And intron splicing, the mechanism that removes introns, is very complicated, and I'm going to tell you more about that in a few minutes. But usually at the end of an exon and the beginning of the next one, there are splice site consensus sequence. There's a donor consensus splice site on the end of exon 1, as I've drawn it, and an acceptor consensus splice site on the beginning of the next exon in line. So the message is transcribed. The introns are removed by this very complicated piece of machinery, which has been studied for 20 or 30 years, and it's still more complicated yet. And the message, the mature message, is translated into a protein. The location then of non-coding variants can be in those 5' or 3' UTRs. They can be located in an intron, and they can be located a very long distance upstream or downstream of a gene in the cis-acting or elements or regulatory elements called enhancers that control the expression of a gene, and they can be a long, long way away from the gene that they control. So as mentioned, there's hundreds of thousands, perhaps a million now, pathogenic variants of hundreds of human genes that cause deafness. And in the drawing on the right, I've indicated in green the genetic loci that have been mapped to particular chromosomes where the gene for deafness is known, and it's likely that the variant was identified as pathogenic. In black font are the loci that have been mapped in families where the variant causing the deafness isn't known yet. And we're going to come back to this issue at the very end of the talk. So, excuse me, the research goals of the laboratory of molecular genetics is to identify novel variants of known genes, novel variants of novel genes that are associated with both syndromic and non-syndromic deafness. And by non-syndromic deafness, I mean only a hearing loss that we know of. And syndromic deafness means that there's hearing loss with other observable consequences. And they could be in the kidney, or they could be in the ovaries, or they could be during spermatogenesis, or they could be in the eye and the retina. And the goal is to understand the pathogenesis resulting in deafness. And the way that we do that is to ascertain large families segregating either syndromic or non-syndromic deafness. The one on the right is segregating deafness inherited as a recessive, where you have to have the variant, the mutation, coming in from both the mother and the father. We often genotype now 970,000 SNPs. These are genetic markers. When I first got started, 300 genetic markers took us a year. 970,000 genetic markers now take us 3 days. So the technology has really advanced significantly in the last few years. So the idea is to try to find a linkage interval on a chromosome so that we're not looking all over the human genome for the variant that causes deafness. And the idea is that in that linkage interval, we should be able to identify a variant that is associated with the deafness. And to study the normal function of the gene, as well as the pathophysiology, we develop usually mouse models, but zebrafish and Drosophila, fruit flies, are wonderful genetic models for studying human deafness, because even Drosophila have many of the same genes that humans have in mice. We study the mutant phenotype using biophysical, biochemical, and cell biological methodologies to reveal the normal functions as well as the pathophysiology. And we think about therapeutic opportunities, both small molecules and, as Ina mentioned, gene therapy. So human deafness genes encode a great variety of proteins necessary for hearing, and I think I've captured most of them on this screen. But what I want to just tell you briefly is that there are proteins that are involved in the actin cytoskeleton. There are proteins that encode unconventional myosins, non-muscle myosins. There are 8 different unconventional myosins in which variants are associated with hearing loss. There are enzymes that are necessary for the functioning of the inner ear, but in many cases, or some cases, we don't know what the substrates are. For example, we don't know what the substrates of CDC14A is in vivo or CLIP, or for that matter, TMPRSS3, which has been studied for now well over a decade. There are genes that encode tight junction proteins. There are genes that encode transporters. There are genes that encode microRNA, gap junction proteins, transcription factors, and a whole host of other functions, which I don't have time to talk about. So the other word that I want to define is in disequilibrium. And what I mean by that excuse me, is linkage disequilibrium. And simply put, if you imagine you have a causal variant in an exon, there may be a benign variant upstream or downstream of the gene, and whenever you see the causal variant, you will also see that benign variant. But it could be the other way around, that the causal variant is upstream or downstream of the gene, and the benign variant is in the exon, in linkage disequilibrium. That when you see one, you see the other. We're going to come back to this in a few minutes. So what's the problem? The problem is that we have ascertained quite a number of families, as have others, where you sequence all the exomes that are known and annotated, and you don't find a causal variant, or you've either missed it, which is not likely, or it's just not in the exome. So you sequence and you find no pathogenic variants. So that pathogenic variant might well be in the non-coding portion of the genome. And the question then is, when you find a non-coding variant that is very rare and close to a gene, maybe in a regulatory element, you don't know if it's causal or just associated with the phenotype. And to help you evaluate these variants, both variants in exons and variants in non-coding region, the entire issue of human mutation in August of this year was devoted, all of the papers published were devoted to evaluating and making predictions about pathogenicity or impact on pre-mRNA or on protein function. And I'm just highlighting two of the papers in that issue, annotating and prioritizing genomic variants using the ensemble variant effector predictor, a tutorial by the first author is Sarah Hunt. And the second paper that I think they're all very good, but this one, Guidelines for Clinical Interpretation of Variant Pathogenicity Using RNA Phenotypes. So these programs are much better than crystal balls. Okay, so now let me start telling you about DFNB39, which was mapped to chromosome 7. And it was mapped by Suzanne Lial's lab, and it was published just about 20 years ago. And it was in a very large region of chromosome 7. And in this paper, the locus was mapped, but the gene and the variant associated in this family was not known. After Suzanne Lial's lab published this, we were in the process of accumulating families that were linked to the DFNB39 interval, which, as I just mentioned, was very large, and there were several genes associated with deafness that are in this linkage interval. So by 2009, between Suzanne's lab and my lab, we had identified 41 families segregating deafness linked to genetic markers on chromosome 7 in the DFNB39 interval. And 20 of these families were quite large. They were all segregating profound deafness. And we were able to reduce the interval to about a much smaller size, about 1.2 megabases. And in that interval, and we could exclude the genes outside that interval, and in that interval, there were only a small number of genes, and one of them encoded for hepatocyte growth factor, which seemed like an unlikely candidate just because of its name. But it turned out that of the 41 families that were segregating deafness linked to the DFNB39 interval, almost all of them had a 3 base pair deletion in an intron of HGF. A few of them had 10 base pair deletion that included the 3 base pairs. So the 3 base pairs were included in the 10. And this just shows you that region of the intron where humans have a duplication of 10 base pairs, the GAT, GAA, and it's duplicated. And if you delete one, you're left with one. Of course, you don't know which one you deleted when you're left with one. So just by convention, the second copy of the duplication is said to have been deleted. And the 3 base pairs is TGA. And the majority of the families just had in that intron the deletion of TGA. And it was a very, very rare variant in control chromosomes, but in the Pakistani chromosomes, there was a pretty high carrier rate. And notice that it's highly conserved, this region is highly conserved, and in the armadillo has exactly the same sequence, GAT, GAA, as humans in this region. So as I said, this region, this intronic region is highly conserved. And what's shown here is the percent of conservation among many, many species. And this is exon 4. And of course, the sequence of exons are usually conserved. And the sequence in other introns are not conserved, but this one is almost as conserved as an exon. So must be doing something important. And that's where the 3 base pair deletion is located. So HGF encodes several isoforms, and we'll talk about those in a few minutes. But the longest isoform is 729 amino acids, and it's a secreted multifunctional cytokine that is proteolytically processed. In other words, it's cleaved, but the 2 parts are held together by a disulfide bond. And it has a signal peptides for secretion, and it has these 4 Kringle domains, which are protein-protein interaction domains. And if you look in PubMed and just type in hepatocyte growth factor, you're going to come up with thousands of papers, thousands, literally. And some of the early papers turned out to call this protein scatter factor when they didn't know it was the same as HGF. And the reason they called it scatter factor is that when it was added to cells in culture, it stimulated them to move around. So it was designated as a mutagen, but it also stimulates cells to divide, a mitogen. It stimulates cells to differentiate, it's a morphogen. It's important for immune regulation, regeneration, wound healing, and as I'll try to convince you of, it is exceedingly important for hearing. So HGF binds to the MET receptor. The MET receptor, too, has thousands of papers that have been published about it. The only ligand for the MET receptor is HGF, and the only receptor for HGF is MET. And this pathway, we've simplified it. It's much more complicated than even this drawing. But what's interesting now is that mutations in other genes that encode proteins in this pathway also cause deafness. So there are variants of the MET receptor that cause deafness. There are variants in the mouse in the Sprouty gene, Sprouty2 gene, that cause deafness. So far, the human Sprouty gene has not been shown to be a human deafness gene, but that's certain to happen eventually. A mutation in GAB1 causes DFNB39 non-syndromic recessive deafness, and they're modified by another locus on chromosome 1 called DFNM1. So this pathway is very important. So we decided to conditionally knock out HGF by deleting exon 5 with the FOXG1 CRE, because it's known that a deletion of this gene body-wide is an embryonic lethal, as is a body-wide deletion of the MET receptor. So you have to conditionally delete it. And when you do, when you conditionally delete it, the mouse is profoundly deaf as compared to heterozygotes or homozygous wild type by ABR analysis. at multiple frequencies. So HGF is important for hearing. There was also a mouse that had been developed and published by Takayama that expressed from a transgene in the presence of normal HGF genes 3 to 50-fold higher level of HGF. And these are images from the Takayama paper, and they have excessive pigmentation. They develop tumors and kidney failure, but the hearing was never looked at. So these are just overexpression of HGF. And what you'll notice is overexpression of HGF results in deafness as well. So it's kind of a Goldilocks situation. You can't have too much or too little HGF, otherwise the mouse will be deaf. And what Risatona showed in my lab is that if you use a RNA scope probe for the full length HGF, or just a smaller probe for just exons 10 through 17, it's expressed during development in the marginal cells of the stria vascularis, which is illustrated on the right, and just at higher magnification, it's the marginal cells that secrete HGF, and illustrated here are intermediate cells and basal cells. And this is just a higher magnification showing HGF in the marginal cells. So while we were working on this, there was an elegant study from a group in California, Shibata et al., in which they selectively eliminated either HGF or MET from mice and looked at the migration of neural crest cells, which migrate into the stria to form most of the cells of the intermediate layer. So in the control, they noticed that neural crest cells approach the stria. You see that here. They adhere to the epithelial layer, and then they incorporate into the intermediate layer of the stria. And when they conditionally knocked out HGF with PAX2-Cre, or conditionally knocked out the MET receptor with PAX2-Cre, the neural crest cells still migrate, and they still attach to the epithelium, but they never move into the intermediate layer. And that's shown at higher magnification here. And what they're looking at is in situ hybridization of neural crest cells expressing the gene, the RNA for dopachrome tautomerase, which is a marker for these neural crest cells. So you can see that the neural crest cells are unable to integrate into the intermediate layer. And that's why the intermediate layer is much thinner in these mice. So far, I've told you that in humans, the del3 and the del10 mutations, deletions of 3 base pairs and 10 base pairs, and HGF intron 4 are only linked with recessive deafness, DFNB39. In a mouse, a loss of or overexpression of HGF in the inner ear causes profound deafness. That incorporation of neural crest cells into the stria requires HGF and MET from the paper from Shibata et al. And that the mouse models corroborate the hypothesis that dysregulation of HGF causes hearing loss, which Julie Schultz published from our lab in 2009. But these two models do not prove that the del3 and the del10 in intron 4 of human HGF are pathogenic variants causing DFNB39. They still could be in disequilibrium with the real mutation in HGF or in an upstream or downstream gene, and we missed it. So the critical question is, are the homozygous del3 or del10 variants in human directly causing deafness? And if so, what's the mechanism? by which these non-coding variants causes deafness? Or are these deletions in HGF, these little deletions in the intron, pointing to the actual deafness genes somewhere else upstream, downstream of the gene? They're in linkage disequilibrium. So to try to answer that question, we can't answer it directly in humans, but we can in mouse by making the same or roughly the same deletion of 10 base pairs in the mouse that causes, that is associated with deafness in the human. So we deleted AA, GAT, GAT, GA in the mouse. And the question is the mouse hearing or deaf? And the answer is the mouse is deaf. So, and what we found, just like in the Shibata paper, is that the thickness of the stria vascularis is reduced. And that's shown here, quantified. The thickness is significantly reduced in the basal, medial, and apical turn compared to the wild type, which are the black dots, and the homozygous mutant are the red dots. Not only that, but the endocochlear potential is significantly reduced. It's not extinguished, but it's just reduced. So I've been talking about this deletion of 10 base pairs being in the intron, but now I have to admit that it actually is in the intron of 3 isoforms of HGF, 2 shorter ones, but it's actually in the 3' UTR of a very short isoform, which we called NK0.5. So is the downregulation or upregulation of NK0.5 the culprit? And the answer to that question is no. So although NK0.5 isoform is downregulated in the 10 base pair homozygous mutant, so is NK1, and so is the full length HGF. So this region is not just regulating the NK0.5 transcript, it's regulating all of the transcripts as far as we can tell from preliminary data that's unpublished. So what we think is that this region of HGF in the intron is probably a locus control region, and it's necessary for regulation of HGF. But why the inner ear is so sensitive to this downregulation, whereas at least in the mouse models, and as far as we can tell in the human subjects, the hearing loss is non-syndromic. So other organ systems are pardoned, but the inner ear is not. And I have to admit that the stria vascularis is not the only problem, the only pathogenesis in the inner ear. The tectorial membrane is a little abnormal, and Reisner's membrane doesn't seem terribly normal, although those are not well studied. So it isn't just a problem with the stria vascularis. But nevertheless, with this 10 base pair deletion, hair cells die and the mouse is deaf. So we think that's the explanation for the human deafness. So now let me turn to the DFNA27 story. So that one too began a long time ago with a project by a postdoc in the lab, Linda Peters, who now has her own lab in New Zealand. And she was studying this large family, and it turned out to be linked to chromosome 4. And so this is the family, the pedigree at the time that Linda was studying. And it's a progressive deafness. Take a sip of water. So the hearing loss is plotted in this graph, and the hearing members of the family are above the 95% confidence interval, and the individuals who are deaf are below that, and it seems to be progressive. And the interval on chromosome, in this chromosome, was about 8.9 megabases. And it was in a gene desert. Basically, that's a big region, but there were only 10 genes. And all of the exons of all of those genes were sequenced. And Linda found no convincing pathogenic variants that were identified. And one of the genes in that interval was REST, that encodes a gene repressor, and we'll talk more about that in a minute. And in 2008, one of the exons of that gene was not annotated in the public database. But there was a paper published in 1999 by a group in Estonia that reported that REST has a tiny additional exon. And we were unaware of this paper, and the annotations of REST didn't include it. But one day I got a call from Botan Bamfi at Iowa and said, "You checked all the genes in the DFNA27 interval, Tom, but what about this little microexon, which is alternatively spliced, it turns out." And it turns out that when you splice in this little microexon, it has stop codons in all 3 reading frames in humans. So it would result in an inactive REST repressor, because the function of REST as a repressor is very much dependent on the sequence of the last exon of this gene. So does human microexon 4 introduce a stop codon? Is there a mutation in this family in exon, this microexon 4 that's alternatively spliced? So let me just tell you, before I answer that question, a little bit about REST. It gets its name from a repressor element 1 silencing transcript. And it binds a sequence upstream or nearby a gene called RE1. RE1 sequence is the REST binding site. And in neurons and in hair cells, the level of REST is extremely low. And it's very low because REST is transcriptionally silenced itself in neurons, but that's not the mechanism in hair cells, which I'll tell you about in a minute. So REST doesn't just inhibit transcription of its target genes all by itself. There are other proteins, and one of the crucial ones that's necessary for REST function is histone deacetylase. We'll come back to this in a couple minutes. So human REST and mouse REST have 2 alternatively spliced isoforms, one with microexon 4, one without. And what Botan Bamfi showed in this paper, in which we were collaborating with him on this REST study, is that if you delete exon 4 in a mouse and make that deletion heterozygous, it's going to make REST, and it makes it in hair cells, and hair cells die because there are certain genes that must be active, not inactive, in hair cells. So the hair cells die and the mouse is deaf. So unhappy hair cells. And they look like this. That is, in the wild type, you have nice hair cells in the organ of Cordy, but in that mouse in which exon 4 was deleted, so that you always make a functional repressor, REST repressor, the hair cells are ablated. They're gone, they die. They form and then they die. So one other question that you might have is, what if you just not delete just exon 4, but you delete the whole gene, gone? And so you have one normal copy and one mutant copy, one deleted copy. And in that case, one copy of a wild type REST gene results in normal hearing, and that's because it's regulated normally in hair cells and REST is inactivated. Well, if you just delete exon 4, hair cells die and the mouse is deaf. But you delete the whole gene and the mouse is happy. hair cells are happy. So now we're back to the question, is there a mutation in microexon 4 of human REST that causes deafness, progressive deafness in the DFNA27 family? So we sequenced microexon 4 in all the deaf individuals of DFNA27 family. And the answer is, unfortunately, no pathogenic variant was observed in the sequence of microexon 4 of REST. So that was initially a little disappointing until Teek Raymond, a postdoc in the lab, looked more carefully at the sequence upstream and downstream of microexon 4. And what Teek found was that in all the affected individuals, there was a G upstream, and the individuals who had normal hearing, and in the database, the consensus sequence, the consensus nucleotide at that position was a C. So again, is this G in disequilibrium with the real mutation, or is it the pathogenic variant? So we have the same question all over again. But one thing that we wanted to know is, are all the individuals with progressive deafness, DFNA27, do they all have one copy of the wild type sequence and one copy with this G in that intron? And the answer is yes, not only for the family that we published, but there now was a new generation, and there are already children who have inherited the G allele and probably will develop progressive deafness. So does that G make a difference, or is it just a benign variant in disequilibrium with the real mutation, which we've missed? So what I've drawn here is that there's a donor site in the end of this exon, a GT, donor splice site, and there's an acceptor splice site in the microexon. And this G in the DFNA27 family, the question then is, does that AG create a novel acceptor splice site? And if so, what does that do to the sequence of this microexon? So before I can tell you the answer to that, I have to mention that, again, that splicing of messenger RNAs is very complicated. It's a machinery with lots of different proteins. And if you are missing some of the novel or important splice factors, you may not splice in a small exon. So here we show splicing of the red exon to the yellow exon, and the green exon was excluded. However, if you also express in a cell a splicing factor called SRM4, you will include some of these small exons, especially in neurons. So SRM4 promotes the neural-specific cassette exon inclusion of some neural-specific genes, and it binds to a specific site where there's a UGC in a polypyrimidine tract. And what's really interesting is that mutations in SRM4 cause the Bronx-Walzer deafness phenotype, which was published by Botan Banthy's lab by Yoko Nakano in Close Genetics. So SRM4 is a critical splice factor for normal hearing. Okay, so what splicing events of REST transcripts occur in when SRM4 is not expressed? And the answer to that is a little more complicated than we thought. Turns out there are two transcripts. There's a splicing from exon 3 to 4, and there's from 4 to 5. And that would include microexon 4 with the stop codon in all 3 reading frames. But there's also a splice from exon 3 to just part of exon 4 and splicing to exon 5, but now it's out of the normal reading frame and it results in a stop codon. And so it turns out that microexon 4 has two donor splice sites, one internal and the consensus one that I mentioned, and you can use either one of them. So what happens when SRM4 is also expressed? And you have the G variant, the DFNA27 G variant, and SRM4 is expressed. So it turns out that there is the use of this now acceptor splice site, because the AG creates an acceptor splice site that the splice machinery recognizes. It introduces some extra sequence, including the wild type acceptor splice site and some additional sequence. And what happens is there are again two transcripts. So one includes 8 amino acids additional, and that's stop codon. And that's a perfectly good transcript in terms of hair cells surviving. So no functional rest repressor in hair cells because the protein translation stops in that microexon. But there's also the culprit DFNA27 transcript that splices using a donor site that's internal, skips the stop codon, splices to exon 5, keeps the reading frame, resulting in a repressor in hair cells at low levels, but enough over time to kill hair cells. So almost towards the conclusion of this talk, I want to mention that the Banfi lab has done a really clever experiment. I mentioned that REST requires histone deacetylase to be a repressor. So what if you use a histone deacetylase inhibitor like SAHA, and you give it at P1 and P3 and P4 to a mouse that is carrying the exon 4 deletion? What happens to hair cells? And it turns out that in the presence of the SAHA, the hair cells look much better. They certainly don't look normal as they do in the wild type control, but they're much better and they actually survive, and there is some hearing. They're not profoundly deaf. And SAHA isn't a particularly good HDAC inhibitor in terms of getting across the blood-brain barrier. There are much better ones than SAHA. So what I've told you in part 3 is that hair cells die when they express full-length functional REST protein. Why hair cells use this strange way of preventing REST from being a repressor in hair cells rather than just down-regulating it transcriptionally is, I think, a fascinating evolution question, because other neurons in the brain just simply down-regulate REST transcriptionally. But hair cells have this kind of unique way of down-regulating it by including microexon 4. So it looks like this point mutation in the intron is the likely cause of progressive deafness. It's not just in disequilibrium with the real mutant allele. It looks like it's the real McCoy, the real mutant allele. And that REST represses target genes in complex with histone deacetylases, and HDAC inhibitors may be of therapeutic benefit for DFNA27 deafness, especially in those young children who have not yet developed hearing loss. Okay, so now my challenge, my slightly megomaniacal challenge to ARO investigators. To date, as I mentioned, there are thousands of pathogenic variants of hundreds of human genes that cause deafness. So what's the problem? The problem is that there are 42 reported human deafness loci for which there are no pathogenic variants of a gene. They haven't been identified, and there are 42 of them. And one in particular, I didn't indicate the date here, but B5 was reported in 1995, which is 27 years ago. So, and others were reported in 2003, 2010, and 2001. So they've been around for a long time, but the variant that causes deafness, segregating in the family or families that were used to map these loci are not known, have not been reported to date. So the failure to identify a pathogenic variant of these 42 human deafness loci may be due to the fact that some of them are bogus loci. And by that I mean they're genetic mapping mistakes, and it's really easy to make mistakes. And it's easy to make phenotyping mistakes if there's an individual in the family that's deaf because they were exposed to an ototoxic drug, but you assume that they have the same genetic deafness as other members of the family. It can really throw off your mapping. And there may be individuals who have normal hearing that aren't, because of their genetic background and other modifiers, are not going to end up losing their hearing. So there are lots of reasons for some of these being bogus loci, but I'm betting that the difficulty is in documenting non-coding pathogenic variants segregating in these families, and the difficulties that we've had in the years that it's taken to figure out the two examples that I told you about. So what I would like to see happen is for there to be an international collaboration to study these 42 loci where the genes have not been identified, the variants of the genes have not been identified. Some of them may turn out to be simple errors, and others may turn out to be really interesting non-coding pathogenic variants, which will take some time, but working together, it might be a fascinating ride. So with that, I just want to tell you the team in my lab who do this work. There are two alpha scientists, staff scientists in my lab, Sayaka Inagaki and Ina Belyanseva, and a group of other wonderful individuals. I want to point out a couple. Takushi Mayoshi has a K99 that was awarded in January of this year, and he's seeking a PI position so he can activate his R00. Above and to the right of him is Brenda Flores, who is starting, sort of spearheading a project to study hereditary deafness in Puerto Rico, and is being mentored by Isabel Rue, who's moving from Andy Griffith's old lab to my lab. And there are two others that weren't in this photograph when we had a picnic outside, Yasuko and Rabia, and a new postdoc in the lab, Samuel Adade. So thank you to all of them. And I also want to acknowledge Mike Ho, who has been instrumental and is the last author on our most recent paper on HGF, and Sheikh Riazuddin, with whom I've collaborated for 25 years, and Rob Morell and Julie Schultz, who spearheaded our first project on HGF and is now at GeneDx. So thank you very much for listening, and I'd love to hear about your questions and concerns and comments and criticisms at this point.
Thank you, Tom. And we have 6 minutes for questions. And I think the audience, people from the audience can unmute themselves and ask questions if it's easier, or type in the chat your questions and I will read it. I don't see questions in the chat yet.
Hi, Tom. Can you hear me?
Yes.
Hey, Tom. Excellent talk. Very, very nice to hear you.
Thank you.
Talk. I had a question earlier in the first HGF. Did you look at the protein levels made the intronic deletions. Excuse me.
You mean of HGF?
Right. When you made the like a 10 base pair deletion intronic region.
So you're asking if we looked at the protein levels. So there are lots of antibodies to HGF, and I'm told still that none of them are specific. So, and there's a person who's worked on HGF all of his career over in the National Cancer Institute. He tells me that none of them are specific. So we haven't looked at protein levels. We have looked at message levels, though, where we can generate a lot of specificity with these RNA-scope probes or primers. So it's a really good question, Kumar, and we'd love to see if we could generate a specific antibody.
Oh, thank you. Just one more question, Tom. In terms of the non-coding sequence, obviously, you did show example of the armadillo and the human, I believe it's conserved. But is it possible that, like in the other segments, you're going to have a lot of non-conserved sequence, making it difficult to use mouse models and stuff like that to model these things?
Yeah, so we were lucky that that region is highly conserved in lots of different animals, including mouse. If you have a non-coding variant and it's not conserved, but it's still extremely functionally important in humans, then it's going to be very hard to nail that down. And you've hit upon probably the most serious issue that's going to confront us when we find variants in non-coding regions, and they're not highly conserved, but functionally really very important, at least in humans. I think that's what you're getting at.
Yes, exactly. Thank you, Tom.
Yeah, thanks, Kumar.
Oh, Tom, this is Anna Laskowski. Again, I appreciated your talk a lot. I was just at the recent inner ear biology workshop in Trieste, and Karen Steele gave one of the keynote speeches or talks, and she kind of laid out kind of an international plan for studying, genetic basis of deafness. And
Age-related.
yeah, if I recall correctly, she said that, we kind of have to come up with like a kind of a standardized plan for taking histories and for, studying the genes. And I don't recall everything that she laid out, but I was very impressed by it. Ina was there also. Maybe she remembers some more of what, some more of the specifics.
Yeah, so Ina was telling me about this, and it sounds like a really terrific effort. And we, of course, will contact Karen Steele if we are able to launch an international project to try to figure out which of those 42 loci are real and what the variants are that are causal. This is probably going to have to be taken up by another generation of researchers.
Yeah.
But maybe we can launch it.
Yeah, thanks.
There are some questions in chat, Tom. One question from Irina Markovich. Regarding project 2, I was wondering if the 10 base pair sequence in the intron that causes hearing loss when detected acts in cis, or if you add it to another part of the intron or
Right.
in another intron.
Right, so we've only looked at homozygotes. That's a really great question. We've only looked at homozygotes. It would be a good idea to look at whether it's cis-acting. We assume it is. But one thing I didn't mention is that we have this 10 base pair deletion on two backgrounds, one with the neomycin cassette inserted, and it's quite a ways away downstream, and one without. And they both have hearing deficits, but the one with the neomycin cassette is more severe. So we think this region is much larger than the 10 base pairs that are deleted, that are involved in regulation. But yeah, we don't know whether it acts in cis or in trans. But we're working on trying to figure out exactly what this sequence is doing at the chromatin level. That's a new project in the lab.
Okay, and one last question. It's a quick question from Jinsei Jung, and he asked, do you think whole genome sequence would be helpful to elucidate the veiled variants?
Yes, that's exactly what we're thinking. You could do a whole genome sequencing and then pull out the exome and look at that, because some of these loci were reported so long ago, they probably didn't do exome sequencing. They may have sequenced a few genes. So yes, I think the idea is to do genome sequencing, pull out the exome, look at that. If you don't find anything, then you can start looking at the remaining 98% of the genome, but we're all just loaded with variants. And it's going to be very hard to figure out which ones are causal. Maybe the idea would be to look upstream, downstream, and in the introns of the known deafness genes if we don't find anything in the exome. But great question.
Thank you so much. I think our time is up, and I would like to thank Tom for the amazing presentation and insightful talk. And I think the rest of the questions we will collect and Tom will answer by email.
Thank you all very much.
Thank you so much, everybody.